Welcome back to this next video in which we are discussing valvular heart disease. Okay, so we've now discussed the cardiac skeleton. Let's now turn on to each of the heart valves in turn and discuss its anatomy. Okay, so we're going to go through them in order. So we'll start off with the tricuspid valve, then we'll go on to the pulmonary valve, then the mitral valve, and then finally the aortic valve. So let me come over here and we'll put our discussion of the tricuspid valve here. So the tricuspid valve first. So the first thing to say about the heart valves is that all of them consist of flaps. Whoops, not tricuspid valves, we've only got one. Tricuspid valve. The first thing to say about all of the heart valves is that they all consist of flaps of connective tissue that are going to shut these holes that connect the two areas. So in the case of the tricuspid valve, it consists of flaps of connective tissue that are going to shut this connecting hole between the right atrium and the right ventricle. And remember, what we are looking at here is just the ventricles. We are not seeing the atria. You can imagine that there is a divide effectively down here. All of this bit that I'm circling now, this is the left ventricle. Here is the entrance to the left ventricle, here is the exit from the left ventricle. And the remaining bit over here, a slightly stranger shape, all of that is the right ventricle. Here's the entrance to the right ventricle, and here's the exit from the right ventricle. We're not seeing the atria, they'd be on top of these holes here. So these are holes connecting the atria to the ventricles, and these are holes connecting the ventricles to the arterial systems. And the heart valves are going to consist of flaps of connective tissue that are able to close these holes, block these holes. And these flaps of connective tissue have a special name. They are called cusps, or you can also call them leaflets. So cusps / leaflets are two key pieces of terminology for these flaps of connective tissue that make up the valves. And the tricuspid valve is very aptly named because it consists of 3 cusps. So its name literally means free cusped valve. So the tricuspid valve has 3 cusps. So let's now see these 3 cusps of the tricuspid valve. Now, remember, when we looked at the tricuspid annulus of the cardiac skeleton over here, it was a very simple annulus, much more simple than the annuli of the pulmonary and aortic valves. It was a flat annulus of connective tissue like so, and the cusps of the tricuspid valve are also going to be very simple. They're just going to be flat extensions of this, so it's very easy to visualize. You're just going to have extensions of this tricuspid annulus inwards that will be able to close this space that joins the right atrium to the right ventricle. Okay, so what are the names then of these 3 cusps? Well, you have a septal one, which I'll begin with. So this one that I've shown here now in green, this is the septal cusp of the tricuspid valve. So I'll just label it up as S. Now, why is it called the septal cusp of the tricuspid valve? Well, the reason is that it is towards the interventricular septum. So remember what I've just said. This part is the left ventricle, and this part is the right ventricle. Now, in between the 2 ventricles, you have the interventricular septum separating them. So you can see that this cusp is quite close to that interventricular septum, hence why it is called the septal cusp of the tricuspid valve. Okay, then the other 2 cusps are in these positions. Here is the anterior one, and here is the posterior one. Now, let me make absolutely clear. These lines down the middle are not connections. The septal cusp is not connected to the posterior cusp. This is a gap in between them. Okay, at the moment it's in the closed position and they're all right up next to each other. But if you wanted, you could imagine that you were a little man standing on top here, and if you were jumping up and down, you could make all of them bend into the right ventricle, and then they'd open up gaps in between them. So these are cuts, effectively. They're connected at the back to the tricuspid annulus, but they're not connected to each other. These are gaps here, holes. It's just that when it's in the closed position, these are right up close to each other, so there isn't actually a space through, but you can push these 2 inwards to open up a gap there. Okay, so I hope that's clear. So there is the septal cusp, and then there is the anterior and the posterior cusp of the tricuspid valve. So it's extremely simple to understand. Septal, anterior, and posterior. Okay, right. So let's now discuss what is the actual structure of these flaps of connective tissue, because there are separate layers of connective tissue that are made up of different types of connective tissue to these heart valves. So what I want to now imagine doing is taking a little sample from one of these. So take a little cut through, and in fact I'm going to do this. I'll zoom in, okay? And then what we're going to do is we're going to cut out, and I'll get my yellow pen, we're going to cut out a little square of this valve. Okay, so cut out a little square of one of the cusps of this tricuspid valve, and I'm then going to have a look at its different layers. So I'm going to draw this here now. So here we go. Brought it out, and here it is. So I've imagined that I've actually, in fact, let me correct this. I've actually got an edge of it. I'm going to take this little cut like so. So we are seeing the edge of the cusp here, this bit here. This is the part that is facing the right atrium above, so this is the top surface, and this is the part that's facing the right ventricle below. So I hope you're oriented to what I'm doing here. So the whole thing is covered by endothelial cells, as always. Everything that's going to be in contact with the blood has to be covered in endothelial cells, and this is certainly going to be in contact with the blood. Blood is going to be up here. Whoops. Blood is going to be up here. That will be the right atrium, and blood is also going to be down here. That will be the right ventricle. And in fact, just to keep us oriented, I'm going to put that this is the right atrium up here, and this is the right ventricle below. So the first thing on the outer side then of this valve is going to be a layer of endothelial cells. So I'll start by drawing those. So here is one endothelial cell with its nucleus. Here's another endothelial cell. Here's another endothelial cell. Here's another endothelial cell. And you get the idea. I will just complete this up now. There's another endothelial cell, etc. And of course the endothelial cells will, as always, be on a basement membrane of connective tissue to give them support. So I'll put that in here. So in red, here is the basement membrane on which the endothelial cells are sat. Like so. Okay, so that's the outermost layer then of each of the valve cusps. And by the way, we're doing the septal cusp here, but they'd all be the same. Right. Now, inside then of that layer of endothelial cells, and I'll just label these up, these are endothelial cells here. Inside of that layer of endothelial cells, you then have 3 layers of connective tissue. Now, I'm going to start off with the layer that is closest to the right ventricle down here, so the bottommost layer. And this is because this is the most thick layer, or the thickest layer. So, big thick layer then of connective tissue. So all of this space is going to be this connective tissue. And I'll just colour this in like so. And this is going to be called the fibrosa, and it's going to contain some fibroblasts. So I'm drawing in a fibroblast here, which are the cells which actually make this connective tissue. Here's another fibroblast. Okay, and this layer majorly consists of collagen connective tissue, so collagen fibres. Okay, so this layer of connective tissue is quite strong, and it's called the fibrosa. It's the thickest layer of connective tissue making up the heart valve, and it consists of a lot of collagen fibres. So collagen is the main constituent, and of course the cells that produce the collagen, those are the fibroblasts. So this is what the fibrosa consists of, collagen and fibroblasts. Then, on top of the fibrosa layer, so this is the fibrosa layer, you have 2 thinner layers of connective tissue. So let's now turn our attention to those. So let's now go above the fibrosa. This next layer here is called the spongiosa, and it also contains fibroblasts. So I'm drawing more fibroblasts here. But now the fibres that they're secreting, the connective tissue fibres that they've produced and which makes up this layer that I'm colouring in yellow here, are different. So this is called the spongiosa, and it's going to consist of connective tissue fibres called glycosaminoglycans. Now, you've probably heard of collagen before. You might not have heard of glycosaminoglycans. So I'm just going to tell you a little bit more about them. So we'll do them over here. Just get a little bit more space. Okay, so the spongiosa, The major type of connective tissue fiber it contains is glycosaminoglycans. And there is an old name for glycosaminoglycans, which is mucopolysaccharides. So both of these words mean exactly the same thing. They refer to a specific type of connective tissue. Now, the name mucopolysaccharides probably tells you something, hopefully, about the nature of these connective tissue fibers. Just before I tell you about the nature of these connective tissue fibers, let me just say that glycosaminoglycans is often abbreviated down to GAGs for short gags. Okay, so collagen is a great big protein. It's polypeptide in nature. You synthesize the tiny starting polypeptides, you make triple helices of them, and then you attach loads of those together to make huge, great collagen fibers. So fundamentally, collagen fibers are made of protein, whereas glycosaminoglycan connective tissue, or mucopolysaccharide connective tissue, is not made of protein. Instead, it's made of polysaccharides, so it's connective tissue made out of sugars instead, polymers of monosaccharides rather than polymers of amino acids. Okay, so what then is the nature of a mucopolysaccharide? Why are we not just saying polysaccharide? There must be some added piece of information here. Well, there is. Mucopolysaccharides, or glycosaminoglycans, have a specific structure. They are polysaccharides that are made out of a repeating disaccharide, and this repeating disaccharide has to contain a uronic sugar and an amino sugar. So, I'll draw a little picture of this. So here are the brackets to show polymer, and then you have two monosaccharides here together in a disaccharide, and the disaccharide is what repeats over and over again. So you have this repeated n number of times. So what I'm drawing here, this is supposed to represent a disaccharide here. Oops, that wasn't very good writing. Okay, so this is a disaccharide. There's one sugar and another sugar here, and you're going to repeat this disaccharide over and over again. And in order for this to be a mucopolysaccharide, you have to have a specific type of sugar in this position and a specific type of sugar in this position. So let me tell you the types of sugar you have to have. So in this one that I'm now colouring in white, this has to be what's called a uronic sugar. And this is a it's a monosaccharide that has a carboxylic acid group in it. So examples would be glucuronic acid. So it's got a carboxylic acid group, a COOH group. And I'll give you an example. So e.g. glucuronic acid is an example. Okay, so that's an example of a uronic sugar that you could have there. This is a monosaccharide. It's very similar structure to glucose, except that in the 6th carbon position, you've got a carboxylic acid group rather than just a alcohol group, as you would have in glucose. Okay, right. So, in the other position, which I'm now colouring in this green colour here, you have to have what's called an amino sugar, which is a sugar with an amino group, a monosaccharide with an amino group. So an example would be N-acetylglucosamine. The glucosamine tells you that it's got an amino group in it. So amino sugars have an amino group, NH2. So an example of an amino sugar would be N-acetylglucosamine. Now, of course, in N-acetylglucosamine, you have actually attached on an acetyl group onto the amino group, hence the N-acetyl, telling you that you've got an acetyl group on the N of the amino group. But it's still got an amino group, and it's still counted as an amino sugar, and it's a very commonly used amino sugar. So, this then is what a glycosaminoglycan, or a mucopolysaccharide, is. It's a great big polysaccharide made out of repeating disaccharides, where the repeating disaccharides consist of a uronic sugar with an amino sugar. Okay, so the different glycosaminoglycans that you can produce will have different uronic sugars and amino sugars that you're using as the repeat. And I should stress that you repeat the same uronic sugar and the same amino sugar in your mucopolysaccharides. So to make this absolutely crystal clear, if you wanted to build a mucopolysaccharide, you need to choose a uronic sugar. Okay, you need to choose an amino sugar. You need to make loads of pairs of them, so that uronic sugar bound to that amino sugar, and then you need to polymerize them all together to have uronic sugar, amino sugar, uronic sugar, amino sugar, uronic sugar, amino sugar. And all the uronic sugars in this great big polysaccharide and all the amino sugars will be the same. And the different types of glycosaminoglycans that you can make will have different chosen uronic sugars and amino sugars. Okay, so this spongiosa there then, it consists of glycosaminoglycans and of course fibroblasts. The fibroblasts are the things which have secreted the glycosaminoglycans. So it's a different form of connective tissue to the fibrosa layer. It's carbohydrate in nature rather than protein in nature. Okay, let's now go on to the final layer, which I'll colour in white up here. So this is the layer that is closest to the right atrium this time. And this will again contain fibroblasts, so absolutely every layer contains fibroblasts, because, you know, this is connective tissue. Fibroblasts are the cells which produce connective tissue, so you always have to have fibroblasts. This layer is called the atrialis layer, because of course it's closest to the right atrium. So this is the atrialis layer. And this consists of fibroblasts, and then what type of connective tissue makes up the atrialis? Well, it's going to be elastic connective tissue fibres. So elastic fibres this time. So we've got these 3 different layers of connective tissue. We start off with the thickest layer down at the ventricular surface, which is the fibrosa layer, consisting of fibroblasts and loads of collagenous connective tissue. Then above that we've got the spongiosa, which consists of mucopolysaccharide connective tissue and fibroblasts which have produced it. And then right up at the top, facing up to the right atrium, you've then got the atrialis layer, which consists of fibroblasts and elastic connective tissue fibres. And we'll discuss the structure of elastic connective tissue fibres much more in detail later on when we want to discuss Marfan syndrome, when we're doing myxomatosis degeneration of the heart valves. However, you will have probably heard of elastic fibres before, so I felt that this was the one I needed to discuss in detail, because you might not have heard of mucopolysaccharides, whereas collagen fibres and elastic fibres, I'm sure you've heard of those before. Okay, right, so that is the different layers then of the cusps of the tricuspid valve, and we're going to see actually that this is the general structure of all of the heart valves, so we can modify this picture very slightly to get the same structure of all the other heart valve cusps as well. But for now we're discussing the tricuspid valve, so each of these cusps of the tricuspid valve here will have the same structure. It'll have this atrialis layer facing up to the right atrium. Underneath that, this thin spongiosa layer, and underneath that, the big, thick fibrosa layer, which faces down to the right ventricle. Right, so there we have it. We have done the structure of the tricuspid valve. Now, to complete our discussion, let's go through how it's actually going to function. So, the resting position for the tricuspid valve is in this closed position. So, at rest, what will if the heart was just off, and you were just looking at it, these cusps, their natural position to be in is the closed position, where they're up in the position that I've shown and they're closing this hole. Okay, they're all coming together, and although there is a little gap in between all of them, they've closed up. Okay, so they're not connected to one another, but they are closing this gap. So that's the natural position for them to be in. When the right atrium above contracts and the pressure of the blood inside it gets higher and higher, what that will do is it will force these cusps open, so it'll push the cusps downward into the right ventricle, and then you'll open up gaps. The gaps here, 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 and right in the centre will open up. So I'm going to try and draw it in the open position. I think that will be helpful. So here is the same hole, and now I'm going to draw it in the open position. So they've now moved downwards, like so. Okay, and this isn't fully open. What you need to imagine is they can be pushed further now downwards, and you can see they're opening up these holes here, and if you push them down further, you'll get even greater opening of the gap between the right atrium and the right ventricle. So now blood from the right atrium can go into the right ventricle. Okay, right, so that's what will happen in right atrial systole. When the right atrium relaxes back down, so it's now ejected the blood into the right ventricle, and the right atrium is now relaxing, so we're in right atrial diastole now, that will now mean that pressure goes right down in the right atrium, and the temptation for the blood will now be to move from the right ventricle below back into the right atrium. What that will do is it will push the cusps back upwards, so the cusp will get pushed back upwards and they'll go back into the closed position. Okay, so that's the way the heart valve works. They can be pushed, the cusps can be pushed down, but then they can't be pushed upwards beyond this closed position. So pressure in the right ventricle isn't allowed to push them, or at least not when they're functioning properly. It's not allowed to push them back into the right atrium, because of course that would open up holes the other way, and then you'd be able to get blood going back from the right ventricle to the right atrium. The final thing to talk about is the fact that there are papillary muscles which help to make sure that inversion doesn't occur when the right ventricle contracts. So let me now discuss the papillary muscles of the tricuspid valve. So, papillary muscles. So, we've just been through atrial contraction and atrial relaxation. Let's now talk about right ventricular systole and right ventricular diastole. So when the right ventricle contracts, it's going to increase the pressure inside the right ventricle, and of course we want blood to be ejected out of the pulmonary valve into the pulmonary trunk, but we don't want blood to go back into the right atrium. Now, when the pressure builds in the right ventricle, there is a great temptation for these valve cusps to invert, to start being pushed upwards into the right atrium. So that's called inversion when that happens. And we don't want that to occur. Now, it shouldn't occur. However, there is an extra precaution to make sure that it doesn't occur, and this is the papillary muscles. So these are little extensions of the right ventricular wall. So they're in the right ventricle, so it's difficult for me to show them on this picture. I think what I will try and do is draw a picture where we are taking a little cross-section through, let's say, here. Imagine we're cutting through here. I'm not just going to draw a line there because I don't want to ruin this picture, but imagine we're going to cut through here, and then I'll try and show you the papillary muscles. So we're going to be looking inside the right ventricle then. So we'll get something like this. So here's the inside of the right ventricle. So we have just cut through here, and I'll make the wall actually have a thickness, like so. So these are the two walls of the right ventricle. We're looking from the front, and here is the actual chamber. This is the right lateral wall. This is this wall here, effectively, and this is this wall here. Okay, now coming out from the wall, there are extensions called the papillary muscles, and I'm going to show these on now. I'll try and make this a little bit more 3D. So here now is the back of the right ventricle here, trying to make it a little bit more, look a little bit more 3D. And you've got 3 papillary muscles coming off here. So you've got an extension here, okay? You've got another one here, and another one here. So these are me trying to show the papillary muscles. So they're little extensions from the right ventricular wall inwards, and they're going to have cords which attach them to the free edges of the cusps of the tricuspid valve. So by the free edges, I mean these edges here. So that's a nice piece of terminology that I'll just add on here. So the free edges of the tricuspid valve cusps are these bits here. So these are the free edges of the septal cusp of the tricuspid valve. These are the free edges of the anterior cusp of the tricuspid valve, and these are the free edges of the posterior cusp of the tricuspid valve. And you actually have 3 papillary muscles, and again they are in effectively the same positions as the valve cusp. So you have a septal one, so this one here is the septal one. You have a posterior one here, and you have an anterior one here. So just imagine these little extensions of the heart wall coming out, a little projection outwards of the heart wall here, okay? And if I show them on this picture here, obviously we are looking from above. We're imagining, you know, the right atrium would be above and the right ventricle is this bit below. So I will show where they are effectively underneath the valve cusps. So here's the septal one, here's the anterior one, and here's the posterior one. So these little extensions of the heart wall inwards. Okay, now from the tips then of the papillary muscles, you then have cords which go and connect their ends to the free edges of the heart valves. So showing this, here they are here. These are these little cords. And it isn't the case that each of these papillary muscles just has cords to attach it to the free edges of its valve cusp that is above it. So the temptation is to think, well, look, we've got a septal one that must attach to the septal cusp's free edge. We've got an anterior one that must attach to the anterior one's free edges, and we've got a posterior one that must attach to the posterior one's free edges. It's more a case that they attach to all of them. So this one might have a cord up to this posterior cusp's free edge. Okay, so all of the papillary muscles connect to the free edges of all of the different cusps. Okay, so like so. And these cords have special names. They are known as the chordae tendineae. So chordae tendineae. And they extend from the top end of the papillary muscle upwards. So here they are in this picture. So here for the anterior papillary muscle here, at its top it will have these chordae tendineae going and attaching to the free edges of the valve cusps above. Okay, so what's the idea with the papillary muscles then? Well, they are extensions of the right ventricular wall. So when the right ventricular wall contracts in ventricular systole, these papillary muscles will contract as well. And when they contract, they shorten downwards. So the direction of contraction, if I get a different colour, is downwards like so. So it will pull this edge up here downwards. It will therefore tighten all of the chordae tendineae, and it will pull the free edges of the valves, the free edges of the valve cusps downwards. Now, why is that such a good idea? Well, this is to prevent inversion. When the right ventricle contracts, this is where we're at most risk of inversion occurring, because now the right ventricular pressure is very high. It will be trying to push the valve cusps back up into the right atrium. But we've got the papillary muscles contracting downwards, pulling the chordae tendineae downwards, and therefore keeping these valve cusps in the closed position, not in the open position. It doesn't pull them so hard that they go into the open position. It's balanced by the push of the blood, which is pushing the valve cusps upwards, but it stops them from inverting. Okay, so the papillary muscles are there to prevent inversion. Right, so there is the tricuspid valve complete. I think we will have a break, and then in the next video we'll discuss the pulmonary valve, and then we'll go on to the mitral and the aortic valves. Now don't worry, it won't take us long to discuss the other valves, because a lot of what we've said for the tricuspid valve is exactly the same for the other valves, so I'll just be able to make small modifications, so it should be much quicker to discuss the anatomy of the other valves.